So welcome back, everyone, and I hope everyone had a nice lunch break. So before I introduce the first speaker, I'm going to just ask Sam and Mike Day, who are online, to say a couple of words, and that will then put the next speaker into context. So Sam, I'll hand over to you.
Brilliant. Thanks, Joe. Yeah, just a couple of words to say. I wanted to first off introduce you to Mike Day from 10x Genomics. Mike will be, and his colleague Kelly will be in a breakout room at the end of the day if anyone has further questions after the talk on 10x technologies. And we're also going to announce a special discount pricing arrangement for UK DRI members on all 10x products, and we're finalizing that at the moment. So if anyone uses or wants to use products from 10x in the future or currently, please get in contact with myself or Mike about that. Mike, did you want to add anything?
You've kind of covered it all. So I just wanted to say hello and say, you know, here at 10x, we're really excited to start working with you guys more collaboratively and more closely. This all started as an idea with Bart about getting 10x products, or our single cell products or spatial products, into your hands to kind of revolutionize and change the work you can do, and really to make 10x as accessible as possible to all of you. So I just want to say thank you firstly for all the talks. They've been really, really great today. So I've been on all day and it's been fantastic. So super exciting to hear what you're all doing, and I can't really wait to see what your plan and the grant proposal, or the grant allocations that go along with this session or this workshop. I think they will yield some really interesting data. So yeah, nice to meet you all, and I'm sure most of you will know me, and if you don't, you know, I think we'll work quite closely together in the future. So yeah, thanks, Sam.
Thanks very much, Mike. I'm going to pass over to Malte now, who is Director of R&D at 10x Genomics for the talk. Thanks, Malte.
Hi, everyone. Thanks for the invite, and good to see some of you that I've been working with previously. So it's great to see you here. Okay, I'll just share screen, I suppose. Do you see my screen and you can hear my voice?
Yep, all looks good.
Confirm. Great. Okay, so yes, my name is Malte Kuhnemund. I am Director of R&D for the In Situ R&D group in 10x Genomics in Sweden. And I was one of the founders of Cortana. And Cortana is a company that has pioneered in situ sequencing. And so I'm kind of giving a presentation that's going to be a hybrid between Cortana work and 10x work. So I'm starting off with Cortana because that is sort of, I want to give you a little bit of an update on what Cortana had been developed in terms of technology until the point that we became part of 10x Genomics now. And then I will give you a little bit of some some outlook on 10x Genomics products, both on what's coming out on the Visium, but also on the in situ, because that's probably what people are most excited about to hear from me, since I'm an expert on the in situ field. Anyway, so in situ sequencing has been a technique that has been pioneered in the Nilsson lab, Mats Nilsson's lab at Science for Life Laboratory in Stockholm, as well as in George's Church's lab in Harvard. In 2013, there's been the first paper that shows that you can actually throw on next generation sequencing chemistry directly on a cell or even tissue sections and sequence very short pieces of cDNA or barcodes that you introduce into the sample. And so sort of like Google Maps, the analogy is to, you know, being able to put a digital layer of information on top of in this case, a satellite image of a city, and you're able to, you know, locate individual houses and what they're doing, the activity of those. And that's, for example, if you zoom into Stockholm, that we would be seeing where Cortana is located on the Karolinska Institute campus. The same idea is to be able to do that for tissue samples. So going down to the single cell level and essentially really mapping the molecular structure of everything that happens inside those cells with single cell resolution, being able to then combine the classic pathology based on morphology of these cells with the molecular information and achieve much higher resolution in terms of classifying different cells in the tissue. Speaking of maps, there's another company that is a spin out from from a lab inside of lab, that is Spatial Transcriptomics. That's the company that was done, that laid the foundation for 10x Visium products. So we're very closely related. We're actually soon moving together in a large R&D hub in Sweden. So just also at that point here, I want to mention that we're really growing the R&D in Sweden. 10x really thinks this is a great spatial hub and maybe one of the pioneering places for spatial biology, and they're really, really investing into this. So we're hiring R&D scientists, and so if you have good PhDs or postdocs in your labs that are interested in an industrial career, let them know that we're looking for really good people. So in situ, what's the strength of in situ, our in situ technique compared to all the other in situ techniques out there, or spatial techniques? Well, it has single cell or even subcellular resolution. You can do hundreds of thousands of genes, but what's really cool is that you actually have the capacity to go down to very short pieces of RNA, but you can really clearly distinguish isoforms and even SNPs. And it offers a very high throughput. So it's already since in 2010, in this paper, the Nielsen lab showed the power of this technology, where it wasn't sequenced yet, but just the power of discriminating individual nucleotides to show in an embryonic mouse, in a sagittal section, where you can distinguish actin beta and alpha actin that are essentially just differing in one single nucleotide. And you can see that here on a single cell resolution, that's neighboring cells. You can distinguish this transcript based on a single nucleotide difference. And seeing this in a mapped out format is just amazing for developmental biologists in order to see how that differs. When I say throughput, the throughput of any in situ technique is really limited by the imaging time you spend on a sample and the magnification that you need to do. So if you scan a tissue section with a, say, 20x objective, which is more than enough for our type of analysis because these signals are very bright, that's sort of how you go around and scan the tissue. So it goes extremely fast. While if you go for mostly single molecule fish type of techniques, you need a very high magnification, which generates a field of view that is this size approximately. And you need to do many of those with a very long exposure time. So it takes typically a lot longer. So the throughput is really a key strength here. So doing biological meaningful studies, you need to analyze many samples in parallel. So that was something that we've helped in this study early on, one of our Cortana's first projects where we helped to map out the neurocrest cell development in embryonic development. And that's essentially the sections that we can do in one experiment, all in one go. So the old chemistry that was published had its limitations. The cDNA-based chemistry was relatively inefficient, and the sequencing like by ligation chemistry had a relatively low signal-to-noise ratio and was kind of limited to 250, which is a good number, but if you want to go higher, we needed a new chemistry. So we really wanted to overcome this lower sensitivity, this lower detection efficiency. And also we really wanted to make it work on many different samples, including FFP. So then Cortana's really contribution in this field is really the chemistry boost that we have developed here in these two reagent kits that we had launched in actually 1 year ago, approximately, in April 2020. That was targeting directly the RNA, which generates a much higher detection efficiency, and then an improved readout chemistry that has a lot cleaner barcode readout, and it's also much faster. So just quickly to explain how it works, if you zoom into an individual cell and you would fix the RNA inside that cell, we then target the RNA with barcoded padlock probes. Those are these circular type of probes that sit down on a transcript and are ligated. And these barcoded probes, these probes carry a barcode that we then later on sequence. You amplify those ligated circles so that these barcodes are essentially single molecules amplified at the original location. And if you then count those, you would count single molecules, right? So it's essentially really quantitative. And then you need to identify what all the different barcodes are, and that's done by the in situ sequencing, apply next generation sequencing chemistry. These spots are then getting stained in 1 out of 4 colors. If you repeat that over and over again, and you sort of remove the color, add new chemistry so that these spots change color, if you then read out the code out of each of these spots, the location of these spots, you kind of follow the sequence of the color code, then you can identify the sequence of that and convert that sort of into a map of digital gene expression. So then every gene gets, identified gene gets a symbol, a pseudo-color symbol, and that's how you can then look at many genes at the same time in individual cells. So that's how it really looks like in real life, the sequencing chemistry. You can then sort of follow these individual dots over a cycle and identify the color code based on that. So the chemistry that we had developed then really has a much higher signal to noise ratio compared to the sequence by ligation chemistry. And it works great on autofluorescent tissue samples because of this high signal to noise. So then how did that perform to our old cDNA, the published cDNA technique, the direct RNA targeting technique? So we took some relatively old archived FFP sections from a melanoma sample, and we then performed cDNA analysis and our chemistry, the in situ sequencing chemistry, and we also applied the direct RNA chemistry on the same consecutive section. So you're looking at the same region, and you see already based on we're just looking at 4 genes here, and based on that already you can appreciate that on a higher level, if you see the same pattern in gene expression that you observe, you just get so much more reads out of that sample. And if you zoom into that small area, that is something you would sort of not really, that structure is kind of lost here. So you essentially resolve a lot more what's happening on a cellular level with this high detection efficiency. Overall, the detection efficiency of that new approach then was up to 12 times higher than before. So we're looking at a much improved detection efficiency. This works then really great on a range of different organs. We've tested that on a bunch of different organs, and especially on FFPE, the performance was really good. So in summary, we essentially achieved a much higher detection efficiency with the new chemistry that works great on FFPE. And it almost actually, we had some samples where we had a sample was cut in half and then freshly frozen, and the other half was put into formalin and then paraffin embedded and sectioned. So we looked at the performance on the same sample essentially, just and it looked extremely similar. So it was very nice to see that it almost has an equal performance on FFPE. It did work. It does work on many different species and organs, and there's also a protein compatibility, which is great. So after you're done with the assay, you can still use the same sample to do an antibody staining. I must say that not all antibodies work perfectly after the chemistry, but it's something that we're also currently improving to make sure that it's really nicely combinable with protein stainings. We have then, since we're part of 10x Genomics, essentially, so that is, I would have to remove this because we're kind of already oversubscribed in the Cortana chemistry access program. So we kind of keep access to those that already had it before. We're not increasing that, but that is because we're also developing a new in situ product, which I'll tell you a little bit about after that. So what you really can do with this new chemistry, this is a 112 gene panel on an FFP melanoma tissue section. So you can really go down to the single cell level and look at the different compartments in the tumor and see macrophages down here and other myeloid cells. You see B cell infiltrating here based on CD3 and CD4, I believe, expression levels. And you see how they are distinguished, how there's fibroblasts separating these two compartments. So that's kind of really the power of the approach here, of the in situ approach. So by now we've really been analyzing many samples. We've done that as a service, but we've also helped many labs and core facilities worldwide to establish the technology. And we could probably say that it's by now the mostly adopted high-plex in situ technology out there. So we've been we've been helping or running it on 25 different tissue types on 5 different species. It works on FFP, fixed frozen, fresh frozen. Already many cancer types that we've been running, and some quite tricky samples as well. And so, and we've already heard probably this morning from Wei Ting and Bart's Rupert's lab, this nice study where we essentially helped out to with the in situ sequencing technology to map plaque-induced genes that have been identified through spatial transcriptomics. So it's a great. It shows really the power of combining 2 spatial techniques here. One to really, on a de novo approach, really looking at what are the genes that are interesting in my sample, but with a relatively limited resolution. And then with our very small subcellular resolution, mapping back in the tissue those genes of interest and getting final resolution. So I just want to point out here the power of the approach again, by being able to apply that on human brain samples, which is fairly tricky because of the lipofuscin. So the older the brain, the more lipofuscin it produces. That's very autofluorescent, so it's kind of tricky to run in situ assays in that sample. But with our technology, we managed to do that. We could quench the background efficiently and then essentially run 222 genes on 3 Alzheimer patient samples and 3 controls. But we just want to show another example where we applied the technology to use single cell RNA-seq informed cell types. And you've probably heard from Kenneth Harris already before, who has essentially perfectionated looking into single cell RNA-seq data to really then make a great gene list from that would identify cell types and then really make a panel for in situ sequencing to map that onto the tissue sample. We've done that for the pancreas. And so in this case, they had single nuclear RNA-seq, and we got a gene list from them to map those cell types back. And it worked really nicely. Pancreas is an extremely difficult sample because of the autofluorescence, but also essentially due to the fact that you have all these digestive enzymes in there. So not very easy to get any RNA or protein assay to work in pancreas, but we managed to really map out all these different cell types in the pancreas. So just some examples pointed out here, some endocrine cells in red, really as they are expected, and some stellate cells up here in green, and the majority of axina cells in blue. Okay, so I want to switch gears here for the last, I don't even know, does somebody tell me how many minutes I have left? I think I have like...
Yeah, you have 9 minutes, 9.5.
Great. I just want to briefly switch gears, which you see based on that. Now I use a 10x Genomics slide template here, and so what is new now? So what do we do here with this third platform? So we have the Chromium single cell tools, we have the Visium spatial analysis tools, and now the third platform for 10x Genomics, the in situ platform. And so the way that 10x thinks about this is to really, use single cell for discovery, spatial to go for translational, and then really going maybe for mapping out a few identified genes more in more detail and on more samples. So maybe going towards clinical in the future. Before I talk a little bit about the future in situ product, which I can't tell you much about at this stage, but what I can tell you about a little bit is some news on Visium. So that the top asks on Visium have been since a while now that FFP is something that needs to be done. So far, it's only been possible to run on fresh frozen and high resolution, obviously. So what's very exciting is that there's the new product coming out for Visium FFP sort of soon. It's almost like in just a few weeks. So I think you can actually start ordering this product soon. I don't know exactly from when. But the really cool thing is that we have essentially developed this product that is now perfectly compatible with FFP samples. And what's been so tricky with FFP is really the RNA, the RNA fixation and degradation. So that you see sort of in this bioanalyzer plot here, instead of having in a fresh frozen, you have very clean, nice, good peaks with RNA quality. In an FFP, you have this very smeary, degraded RNA. So that's been what causes so much problem. So the new product is really similar in terms of the workflow. You section your FFP tissue sections onto the VCM arrays. You do an H&E or an immunofluorescence staining. You can then do the imaging on that. And then there's the FFP VCM workflow, very similar to the normal VCM workflow. You do the library prep and do sequencing. And what's been amazing, and this is actually an achievement that is primarily run out of Stockholm, so the R&D site in Stockholm to develop this technology, is that it really almost performs similar to the fresh frozen VCM. So you see the number of genes detected here and the genes per reads per spot. So it's extremely similar in performance, which is fantastic. And so then you can do this type of analysis on tumor samples and really ask those questions: are there any immune cells infiltrating and so on. The other thing is the increased resolution. That's been a major ask, and so we are working on something there, and it looks very good, and it's going to be coming by mid or in the first half of next year. So it's not that long until this product is going to be out, and it's really to go down to the single cell level with VCM. So the first generation that was essentially developed by Spatial Transcriptomics back then had around 100 micrometer in diameter and quite a lot of dead space between the array spots. Then VCM was a major improvement with those 55 micrometer spots, but much denser, so you don't really miss out too much. But then VCM HD really is kind of a revolution here. So it's essentially an increase to the first generation of 1,500-fold in resolution. So then really going, so you can sort of appreciate the change in resolution from the size of the VCM spots here. You'd sort of see there's a couple of cells per spot, 5 to 10 cells maybe. And if you look at that in VCM HD, you'd sort of see that, yeah, you're covering like maybe less than 1 cell per spot here. So that's something coming out in the first half of next year. And then quickly, just the last 1-2 minutes, the In Situ platform that is developed now in 10x is based on the foundations of Recor and Cortana. And the idea is really to offer a turnkey instrument with optimized chemistry. It's going to be different from the current chemistry. It's going to be much better. There's going to be a high-end software and visualization tool that's part of the offering. And you will be able to do RNA and proteins combined with obviously subcellular resolution. There will be pre-designed panels, and we will also offer custom gene panels. But that's unfortunately all I can say about the new platform, but you just have to stay tuned and see what happens so that we will be able to show you more things soon on that end. But thank you all. Thanks for inviting me to this conference. Thank you so much. Listen. Great.
Thank you very much, Malte. So we have one question that's come in first off, and this is from Bassam Hassan, who says, very nice, thank you. Are there any limitations on what type of fixation, embedding, or thickness of human brain tissue that can be used in your pipeline? And then as a follow-up, how can people get in touch with you to discuss projects?
Yeah, okay. So limitations, I think one limitation is the thickness of the tissue. So we typically go for 10 micron. 20 is fine still, but much thicker than that we can currently not do. So that's one thing. In terms of size, so currently with the Cortana chemistry, we can analyze relatively large tissue samples, so even up to 4 square centimeters, so like 2 by 2. Yeah. So anything that kind of fits on a standard microscope slide. For the future in situ product, I can't really tell you right now what that will look like. But other than that, there's no bigger limitations than that. I would say what's definitely key, even though we have good data showing that even if we have relatively fragmented, degraded RNA, the better your RNA quality, the better the results will be. So if you check your RIN values of your samples, the RNA quality, yeah, that's definitely a limitation, which is the same case for almost any technique.